Hello, and welcome to this video on nucleotides for signal transduction. At the end of this video, you will be able to describe how GTP is used to induce allostery in G proteins and describe how phosphates transferred from ATP are used to change the chemical properties of a molecule. GTP is a key molecule in regulating many proteins called G proteins. Remember, first of all, that GTP is a nucleotide. It's guanosine triphosphate. So the base out here is guanine. It's got a hydroxyl group here because this is one of the RNA nucleotides, and it's got 3 phosphates. So it's guanosine triphosphate. GTP binding flips the switch to activate G proteins using allostery. So G proteins are a special class of proteins. There are several kinds, not all of them are used in signaling. And the thing that makes them G proteins is their ability to bind to GTP. And essentially these G proteins have 2 states. They can be on when they're bound to GTP, or they can be off when they are bound to GDP. That's the diphosphate version. It only has 2 phosphates. And these proteins can switch between these states by either adding GTP or by hydrolyzing GTP back to GDP. So let's go around starting from the off position. The protein here is bound to GDP, that's the diphosphate version. Now remember that GTP is a high energy molecule. It is made like ATP through a series of high energy processes. And so these G proteins don't really have the ability to make GTP. So what's happening here is not the addition of a phosphate to GDP. Instead, this is an exchange, and it's facilitated by this protein called GEF, the guanosine exchange factor. So what happens in this activation process is that GDP leaves, and GTP, which was made somewhere totally different in the cell, enters and binds. And that causes an allosteric change and activates this protein. Now it's in the on position. Again, this is not phosphorylation, it's an exchange. GDP leaves, GTP comes in. Just like you were exchanging a shirt that's too small, and you give one over and you take a new one. That on protein can be turned off through hydrolysis. So creating GTP is a positive delta G high energy reaction. The hydrolysis of GTP is negative delta G. It's spontaneous. You don't have to add any energy to do it. And so when we go from the on to the off state, we use this protein, the GAP protein. And the GAP protein stands for the GTPase activating protein, where the P here stands for phosphatase. A phosphatase is a molecule that removes phosphates. And in this case, it's removing the phosphate from GTP, which has 3 phosphates. This phosphate is being lost, and you end up with GDP. And again, now the protein is bound to a different molecule. Now it's bound to GDP. And so it undergoes an allosteric conformation and now is back in the off position. So again, G proteins can cycle between the off and on conformations by either adding GTP or by hydrolyzing that GTP back to GDP to turn the molecule off again. We will see many proteins that work like this. Some of them are involved in signaling, and later we'll see a number of other proteins involved in other processes. ATP is also a key molecule in signaling, and in this case, it is not just providing energy. Remember that ATP is also a nucleotide. So here's your adenosine out here. We've got your hydroxyl because this is an RNA nucleotide, and your 3 phosphates, that makes this ATP. This is the same ATP that's used in the production of RNA, the same nucleotide. And it's also the same ATP that's built by ATP synthase and used as an energy carrier around the cell. Here we're going to use it for a third function, which is giving up a phosphate to another molecule in order to change that molecule's chemical properties. So ATP can donate its 3rd phosphate to flip the switch by changing the chemical properties of a recipient molecule. And that looks like this. The proteins that move phosphates from ATP to a recipient molecule are called kinases. This is one of the few enzyme names that isn't just intuitive. So it's worth remembering that kinases move phosphates from ATP to another molecule. Again, ATP is made via the electron transport chain. It cannot be made here. We're not in the electron transport chain right now. So those ATPs are made somewhere else. They're brought over here. And this protein that is off can be turned on by taking the phosphate from ATP in this negative delta G ATP hydrolysis and giving the phosphate to this new molecule. And now that new molecule is in the on conformation. Remember that phosphates are very negatively charged. So when we look at phosphates, they're very polar. And they have a lot of negative charge. So in adding a phosphate to this molecule, what we've done is add a set of negative charges, change the potential for chemical interactions in that protein. It's easy enough to undo that simply by using a phosphatase, which is a protein, again, that removes phosphates. And so by removing the phosphate from that blue protein, we take it from the on and active conformation back to the off conformation. And that phosphate that's removed can be transferred back to ADP, although that's really high energy and unfavored. Instead, it's probably going to exist just like this as inorganic phosphate floating around in the cell. So what we've seen here is that ATP and GTP can serve as intermediates in cell signaling by turning proteins on and off. And in doing that, they connect the cell signaling pathways to nucleic acid biology and metabolism.